name is Alexandre Marra. I'm a professor of the postgraduation stricto sensu program in health science at the Albert Einstein Instituto Israelita de Ensino e Pesquisa, and also a researcher at the Hospital Israelita Albert Einstein. I have been research at the University of Iowa Internal Medicine Department in Iowa City, Iowa, for 6 years. I'm really excited to introduce the sixth presentation in the Einstein Connection series of guest speakers. Einstein Connection is our international program that intends to connect Brazilian researchers, students, and healthcare personnel with international professors and international researchers. It's my pleasure to introduce our guest speaker, Professor Gen Shinozaki. He's the Associate Professor of Psychiatry and Behavioral Sciences, Division of Medical Psychiatry at the Stanford University School of Medicine. Professor Shinozaki is also a member of BioX and Tsai Neurosciences Institute. He has also been mentoring many graduate and undergraduate students at the University of Iowa and Stanford University. I have been honored to have Dr. Shinozaki as a mentor for my son Pedro at the University of Iowa, and it's been rewarding to see Pedro collaborate with Dr. Shinozaki on a couple of research projects. Dr. Shinozaki will talk to us about from delirium to the epigenetic investigation, the successful story of a research led by a physician scientist. We have a plan for Professor Shinozaki to speak for 40 minutes, after which we will have 10-15 minutes to answer questions. You can unmute yourself and ask your question after Dr. Shinozaki's talk, or you can type your question in the chat. I'd like to ask all of you to answer the survey at the end of the presentation. Thank you, and I will now hand over to Professor Gen Shinozaki to start. Thank you.
Hello, everyone. Dr. Marra, thank you for your kind introduction, and thank you for giving me this opportunity. And I'm very excited to be able to talk with a young scientist from Brazil. And as Dr. Marra mentioned, I had been honored to work with many bright mind undergraduate student, including his very smart son, Pedro. And many of work I'm presenting today is a collaboration of me and him and other students. So I hope you enjoy. Let me share my screen. And I hope you can see this. Okay, good. All right, so let me start. So I put the title from delirium to epigenetics investigation. And I also put our EEG device development work related to delirium. So let me begin with delirium EEG device work, then followed by epigenetics work. And then hopefully we can discuss how we work as a physician scientist in this academia and the science. Okay. And I have already published a couple of papers related to our epigenetic, EEG device development work. So after hearing my talk, if you become interested, please take a look at those papers. Okay. I like to start my talk with this question. Why do we measure blood pressure? Or how about glucose? Why do we measure those? Any volunteers? I doubt. It's a little bit hard to speak out in front of many people. So I'd like to go ahead and answer this question. We believe that we measure those blood pressure or glucose because we want to identify patient at risk. Like, for example, if we find a patient with high blood pressure over 200 in systolic blood pressure, we feel that patient are at risk for something bad happening. And if patient glucose is too low, we are worried about hypoglycemia. The patient may die. So we need to respond quickly. I want to come back to this question later. So please keep them in your mind, okay? Then next, I want to borrow your imagination. let's say your grandma or grandpa or your mother or sister, depending on your age and generation, if your family, a loved one, was admitted to the hospital, you would hope that he or she would come home safely, right? But it's not that easy because there's a condition called delirium happens many times among elderly patients. So we know that aging and the delirium is strongly related. So we live in aging society and everyone is aging, including me and you. But many literature says that delirium is common and also dangerous. But at the same time, many literature says that it's underdiagnosed, and that's why it's undertreated. So there has been a lot of effort to improve how we can detect delirium, but current methods or instruments are unfortunately not practical for screening or detection. And because those are not practical, or it's not practical, it's not widely used in clinical practice. I said that delirium is common. How common is it? It's like 12 million elderly people over 65 were admitted to the hospital annually in the US. Among them, 20 to 50% in general medicine floor, or 15 to 53% after surgery, or close to 90% of ICU patients develop delirium. So in total, at least 2-3 million cases of delirium happen every year in US alone. And I think it's same for US, Europe and Japan and other places. I said that delirium is dangerous because delirium patient can have a higher incidence of complication like fall, or injury or infection. And because of those complications, they tend to stay longer in hospital. And some research said that they tend to stay like more than 1 week. And because one hospital stay cost, one day hospital stay costs over $2,000. If you stay longer for 1 week, that can easily add up to $15,000 more. And the most problematic issue is the high mortality. It says that after delirium, up to 40% people die after 1 year, which is very severe case. And more likely to be discharged to facility like nursing home or rehab place. And because of that, overall cost for delirium is said to be $60,000. So because, as I said, there are 2-3 million cases of delirium case every year, the total cost for healthcare system can be $150 billion. That's a lot of money. And Dr. Sharon in Norway, as I put references here, published a lot of paper like delirium in older person in New England Journal of Medicine, or delirium in elderly adults in Nature Review Neurology, or delirium in elderly people in Lancet. I mean, major journal, but almost the same topic journal, the same topic article has been published, meaning that Dr. Inoue tried to increase the awareness of this issue, but nothing has changed, unfortunately. As I said, after delirium, people die pretty quickly, like up to 40% die after 1 year. So there's a lot of effort, as I said, like current screening tools, including CAM-ICU or DRS, and more non-specific point of test includes MMSE or MOCA. But the problem is the human beings need to administer them. This is the example of CAM-ICU. It's nicely done with colorful chart with the hope that it's easy to be used by nursing staff. But the problem is nursing staff are busy to use it. That because of that, it's hard to be implemented regularly, consistently. Then I thought about EEG. I think I skipped. So I thought about EEG could be good too, because traditional EEG can detect delirium. But what the challenge of the traditional EEG is, it's not practical for screening for every patient, even limited for high-risk group, because the machine is too big. The technician needs to put electrodes all over the head. And finally, neurology specialists need to interpret that. So it's hard to use it for every patient every day. However, the EEG findings for delirium is said diffuse slowing, meaning that slow wave like delta or theta can happen across all 20 channels diffusely. Then if every channel is showing the same signals, I thought we don't need to use 20 channels, but just a few channels is enough. That was my thinking. So this is an example of diffuse slow wave. This is an example of normal EEG. And if you compare right and left, you can tell the difference, normal and diffuse slowing. Then even if you take only 2 channels, you can tell the difference. The first one is slow, and the middle one is normal signals, and the last one is slow. So you can tell the difference just by 2 channels. You don't need 20 channels. And in fact, this fact has been known since 1940s. This is a famous paper published by Ingel and Romano. The first one, top one, is showing marked delirium, showing the slow wave, and the below one is a normal awareness, like showing regular frequency. And this is obviously just for 2 channels. And this technology has been used in other areas, actually. In the area of anesthesiology, they have been used to monitor the depth of anesthesia. The machine is called BIS monitor or entropy. My understanding that this has been become the standard of care for anesthesiologists. And even in our own area of psychiatry, there's a machine called ECT, electrical convulsive therapy, which is giving electrical power to the forehead to induce seizure for the treatment of severe depression, which is very effective. When I was a resident, I saw that machine, and I realized that we are using the few channels EEG to monitor the duration of seizure activities. Then I thought that I should use this machine to detect delirium, but the machine itself was still too big to carry around. So this is an example of BIS monitor. So you can put the electrodes on the forehead, then within like 30 seconds, you get this score like 98. Then anesthesiologist monitor this number to make sure that this number is kept below 50 to make sure that patient do not wake up during the surgery, which is really bad thing. So I thought this machine could be good, but at the same time, this BIS monitor is too big to carry around in the hospital. So the concept is, do you want to use this kind of 20 channel or this kind of small device to detect delirium? And I think answer is obvious. So I did an EEG study working with my student and recruited over like 1,300 subjects over 3-4 years in University of Iowa. And roughly 30% was a positive patient with delirium and 70% was a control. And roughly 2/3 came from general medicine, 20% came from orthopedics, and the rest ER from ICU. The male and female were half and half. Average age was 68. So this was the machine we used at the beginning, like palm-sized EEG device. They put electrodes on the forehead like this. And when we record like a right and left 2 channels, for the sake of data gathering, we start with 10 minutes first. Then from raw data, we converted to like spectrum density analysis. Then we invented some algorithm to capture good signals. So this is the first case, like the black one is a normal case without delirium, and the green one is delirium cases. Then when you see that there's those two patients were totally different, separated, which was good. But not only that, when the delirium patient became really delirious on day 2 and day 3, the score picked up. Then when patient recovered, went down to normal range. So that was very nice start. For your reference, this y-axis is the BSEEG score. Then higher score meaning more slow wave. Okay, but because this was a successful but one-to-one case, so I need to do more. So that's what they did. When we did 45 samples in the positive group and negative group, we were able to see the difference. And with cutoff, the AUC was 0.70, which is not the perfect score, but it's a good score. Then sensitivity is especially over 80%. But we need to validate and replicate, and that's what we did next. With additional 24 subjects, AUC was 0.8, and again, sensitivity especially was over 80%. And when we brought this to ER, over 100 subjects, AUC was again 0.84, and the sensitivity especially roughly 80% around. Then we also tested with a smaller device. I call it the sum-sized EEG. And we again confirm that DRS is delirium rating scale. So the x-axis is the severity of delirium. Then BSEEG score is like a y-axis. Then we can see the dose-dependent increase of BSEEG score in proportion to delirium severity. Then so far, I showed that EEG can capture delirium, but why do we care about delirium? Because delirium is associated with poor outcome, like extended length of stay, or death, or disposition not to home, right? So my next question was, can EEG predict what's going to happen in patient outcome? That was my next question. Because, delirium is associated with poor mortality. So first, we want to make sure that our clinical team evaluation is replicating what's known that people with delirium die faster. And we did a nice job with replicating that. But the real question is, can EEG do the same thing? And it did. The EEG score, BSEEG score high, meaning more slow wave, compared to BSEEG low, like less slow wave. And you can tell the difference of their outcome, mortality, from actually day one EEG, meaning that the first EEG score when they came to the hospital. Instead of clinical evaluation, our EEG device can predict what's going to happen 1 year after they come to the hospital by only one time EEG, even after controlling for other variables. So this is a good comparison, clinical category and EEG category. So technically, we can replicate what psychiatrists or human being can do with just simple EEG decoding. And the nice thing about this EEG scoring is different from yes or no, delirium yes or no, dichotomous differentiation. This is a more continuous variable like blood pressure. So we divided the delirium BSEEG score into high, middle, and low. And then you can see the dose-dependent high risk for mortality. And now this has been replicated over 500 subjects and more. So let's say your grandma in the hospital, then you'd hope that she'll come home safely. Then we know that we measure blood pressure or glucose. Then my question for you would be, why not BSEEG? If we know that BSEEG can detect high-risk patient, we should start using this in a future clinical practice in the hospital. So the future direction, I envision this BSEEG score to be used as a next vital sign, like used every day and every patient in the hospital and clinics and nursing homes. And this some-size new device with iPod app is under development. So hopefully we can bring this to the bedside for our patients. Okay. So far, this is the end of the EEG story. Should we take some question now, or maybe we can go to the next topic of epigenetics?
I think that you can go next because that's perfect.
Okay, all right. So I'll be happy to take a question for EEG next after the end of this talk, okay? So let me go to the next section, epigenetics of delirium. So also I published a couple of papers about this, so please review this if you're interested. So epigenetics, I'm not sure if you are familiar with this, but we know that genetics, we have a DNA in our body, and DNA sequence doesn't change. But we know that there's a modification on the top of genetics, and that's kind of what we call epigenetics. There's like a different meaning of it as well, but let's make it simpler. And there's a different mechanism called in epigenetics, including the DNA methylation. Not only brain, but we use the back sorting glia and a neuron and blood. Then you see that glia, every single CPG has a negative correlation, whereas the neuron do not show that. So this was very consistent with what I described. In microglia, these cytokine genes methylated less methylated along with aging. That's why they can be primed to release cytokines more easily when you get older. And this is more responsible in glia, but not in neuron. Okay. Then you may say that this is not about delirium. So that's why, yes, I agree. Then we need to compare a case of delirium and the control. So that's what we did next. So this is, we compare the 43 delirium patient and the control patient, and the demographics are pretty much equivalent because we matched. Then we initially looked into TNF-alpha gene, as we know that this is a significant difference between along with aging. Then when we compare the delirium case and non-delirium case, and again, a TNF-alpha gene, again, we see the stronger correlation of negative correlation with age among delirium case and not in control. So this is again consistent with what we predicted. People with delirium are more prone to express cytokine, pro-inflammatory cytokine among their blood. As you see in the picture, the red one is delirium, so like a steeper correlation and correlated, whereas a green one, the non-delirium case, there's no significant correlation. But because we did an array method which covers genome-wide data, instead of focusing on just cytokine genes, next we focus, we try to analyze rest of the genome-wide data, what we call it network analysis or enrichment analysis. What do I mean by that? Meaning that you get several signals of gene hits from the analysis, but you need to understand how each gene interact to make sense of biological function. So that's what we can do as a using bioinformatics tools. So this is example of GO analysis. GO stands for gene ontology. And the top hit was, to our surprise, very relevant signal, immune response or cell activation. And those are even like significant after multiple comparison false discovery rate. And another network analysis called KEGG, Kyoto Encyclopedia of Gene Graphic, or something like that. And then again, top hits, one of the top hits, including cholinergic synapse, which is very relevant for delirium pathophysiology. So the demanding challenge for this sample was those patients were recruited from various hospital settings, including, as I said, general medicine, orthopedic surgery, or ICU and ER. But etiology of delirium can be diverse. So DNA mutation signal may not be generalizable, so the signal we found here. So to address that, we need to do a setting called post-operative delirium, before and after surgery. Because for surgery patient, the insert is the same, then we can deconfound the etiology of delirium. So when we use a neurosurgery sample, we use the before and after sample, and then focus on post-operative delirium. Then sure enough, when we did a GO and KEGG analysis, enrichment analysis for these samples, although sample was limited to like 19 subjects, the top hit was cytokine production, TNF activation, defense response, and the cytokine interaction. This was very significant, and I was very excited. And the more data is really confirming and replicating. So summary, the DNA mutation level at the CPG in TNF-alpha gene decreased with age in blood sample from large cohort, and also confirmed the expression level increased with age. And also we found in glia in human brain, but not in neuron, and also in blood from delirium patient, but not from control. And also we confirmed other methods to confirm the replicate that. So GO analysis indicate the role of immune response, inflammatory response, and cytokine production, and TNF activation. And the KEGG pathway showed a cholinergic synapse and cytokine-cytokine interaction. And DNA mutation panel showed promising AUC, which I didn't include today. And actually, this signal has been replicated 4 independent cohorts. So we are trying to publish this data now. So hopefully you will see it. So stay tuned. So conclusion, we show the DNA mutation difference both at gene and network level between delirium case and control, and evidence in inflammatory related genes. And this finding indicates that DNA mutation status in blood may become a useful epigenetics biomarker for delirium. Epigenetics process may play a key role in pathophysiological mechanisms of delirium. The future direction, I'm hoping that pre-post neurosurgery sample comparing ongoing. We are trying to replicate samples. And we are obtaining more independent samples. And brain tissue analysis is ongoing. And actually, we are developing a mouse model to investigate the medication effect on DNA mutation signals. So I put the last slide, peri-operative protocol in 2030 maybe. So we collect the blood from the patient and analyze DNA mutation data, and then risk prediction, and also we obtain baseline BSEEG score. And then after high-risk surgery, we collect another sample and also monitor BSEEG for risk monitoring. Then we can monitor who are at risk, and we can capture pretty quickly when they develop delirium. So this could be a future practice for surgical patients. The bottom line, we want to have a happy grandma back home. So that's why we are working on this. Getting back to the how I developed this career so far, I think it's all depends on thanks to my mentors. And I met many mentors through medical school. Dr. Kamba was a psychiatry chair at my medical school. I was very young, like 30 years ago. And then followed by Dr. Marra, who was a professor at Mayo, and who guided me. So those two are like Japanese mentors. And when I started my residency at the Mayo Clinic in 2004, which is 18 years ago, Dr. Ken Philbrick was a program director. He was almost like a close to God, and he is very generous and kind. And he is the most trustworthy gentleman I've ever met. And now I call him as my friend. So I'm very honored to be his mentee and a friend. And then David Mrazek was the chair of my Mayo Clinic residency program. And he somehow decided to pick me as a mentee and start supervising me. And he introduced me to the area of psychiatric research, starting from pharmacogenomics. And unfortunately, he passed away from glioblastoma, which I was very sad, but he really opened the door for me. And Jimmy Potash was my chairman at Iowa, who recruited me after my J1 waiver job. And he guided me through academic career. And he was originally from Hopkins, but a couple of years ago, he went back to Hopkins as a chairman over there. So I'm glad to see he's going up the ladder to the higher position. But at the same time, I thought it's a good time for me to be independent. So I stayed in Iowa a couple of years and moved to Stanford last year. And so I believe that, finding great mentor to work with who truly care about you is very important for your career. And as you see, I have been very fortunate. I have many strong and kind mentors. And at the same time, science cannot be done by yourself. So you see the number of collaborators I have. These are the collaborators when I was in Iowa, and actually I'm building a new network of collaborators in Stanford as well. But I had a collaborator from every single department in university, like surgery, medicine, orthopedic surgery, neurology, pediatrics, neurosurgery, and pathology, and even dentist, and ER, and family medicine, biology, and all over. And they help me a lot because I can't do everything by myself, obviously. And you need to find a good collaborator, and you need to be open-minded for new opportunities. And again, I have been fortunate with many students joining my lab. I started with like one student, of course, like 2014, then quickly it became a big group. And I was able to mentor a lot of students, undergrad who went on to PhD school and medical school, and also many medical students went on to psychiatry for their residency. So at least after working with me, they didn't change their mind to become psychiatrists, which was a good thing. And this is Pedro. Pedro has been one of the brightest students I've ever mentored. And he had won a poster award from a national meeting last year. And this year, he was awarded a prestigious excellent in undergraduate research at the University of Iowa. And I think he published more like 6 or 7 papers with me already. And now he is drafting a very first author paper of his own. He has already one co-first author paper. And we are having fun. Research is fun. It can be tough, but as a group, we can put our talent together and we can do a lot of good things. Okay. See, somehow so then I moved to Stanford at a new adventure. So I moved with just one postdoc, but now I got 3 postdocs and 5 undergrads. So I'm building a new team again. All right, thank you very much. And so I'll be happy to take any questions.
Great to talk to you, Dr. Shinozaki. I really appreciate, and I will open to questions. But I have a lot of questions. I did a lot of notations, and I'm really, really impressive because you are a psychiatrist and performing, studies in a different broad, like animal model to human model. I enjoy to see how you describe the delirium and the comparison to glucose and the blood pressure, because delirium, principally in ICU, is really high, more than 40%, as you showed for us. And the metric is sometimes very subjective, and you try to find biomarkers to answer these questions. And I have a lot of questions, but I'm really interested in this EEG device, this small one that you showed us. My question is, do you have a patent about that? And if yes, how your advice for the young researchers when they have your research questions, and how to protect your ideas as you are doing here?
Yeah, so that's a good question. And I did apply for patent, and actually a couple of different patents. And the reason for that is when at Iowa, University of Iowa encouraged us, like faculty and students, to seek intellectual property. And I did have a chance to utilize their mechanism. So there's an office to encourage and manage those intellectual property invented by faculty and staff and students in Iowa. So through that assistant, we were able to work with lawyers they hired, and then we filed for that. It's still pending approval, and who knows what happens. But that's a great opportunity. And through that, I had opportunity to run to think about a commercialization of the technology. And I believe that, which has not been successful, unfortunately, but hopefully someday. But I realized the importance of bringing the scientific discovery to the bedside. Because as I said, I'm a clinician and a psychiatrist, and my hope is to be helpful for the patient. And I do believe that my research is really directly beneficial for the patient clinical care. But to do that, probably successful commercialization is a key. And I'm hoping to devote more effort on that side because I moved to Bay Area. So hopefully I can find some good investors and startups, venture capital to support my endeavor. So we'll see.
Wonderful. And considering this new device, considering to be commercialized, who is taking care of this process? You by yourself or someone is helping you at the university?
Yeah, so university has an office who talk with lawyers, and then they would take majority of the task. But sometimes they they decide to carry on, or they decide not to carry on, because IP process legal fee is pretty expensive. So some of the patent they release to me, so I'm dealing with by myself, which is very pricey. So I need to find a good business partner to deal with. So that's a challenge as well. And also you can apply for NIH grant, NSF grant that is specialized for those commercialization as well. So that you can do. I have been trying it, so far no success, but you can try.
Wonderful. Hey, this is part of the process now, because it's very innovative thing, and I appreciate that you share with us your thoughts about your research and these new developments that you are doing at bedside. And I'm totally share with you the same thoughts that we need to do this to improve, the care of our patients. I think that's a very reasonable thing. A very fast press about your big network that you build during your entire life. And my second question is, I've heard about that you have a mentor in Mayo Clinic. I think that you were there before moving.
Yeah, so I did a residency at Mayo.
Okay, did the residency in Mayo, okay. And that's not my question. What was it like to live in different US states? Which are the positive aspects or the negative ones that you can share with us? Because I understand that's pretty interesting, like to do these movements, but how this is good or not for your research, because you need to build another lab, and I'd like your thoughts.
Yeah, so far, so I'm originally from Japan, and I moved to US for my residency. So I moved to US like 2004, and I did residency for 4 years and followed by 1 year fellowship. So I spent 5 years in Rochester, Minnesota, Mayo Clinic. Then after that, because I came with J1 visa, I need to do J1 waiver job. And my wife started her internal medicine residency at the University of South Dakota, Sioux Falls. So I found a job, was there as a staff psychiatrist at the VA in Sioux Falls, Veterans Affairs Hospital. Then I moved to University of Iowa. Then spent 8 years. Then at the University of Iowa, I spent more than 50% of the time I have been doing research. Most of the time I was doing 80% research. And now I'm back to 80% research again in Stanford. So far the Mayo, Sioux Falls, and Iowa, and Stanford. So 4 institutions I worked in the US. And surprisingly, there's a lot of course, there's a lot of similarity, but there's a lot of differences in each institution. And some are stronger in research, some are stronger in clinical aspect. Some are so for example, Sioux Falls, it was very hard to do research, obviously. So that's why I chose University of Iowa to go back to be in a strong academic institution. University of Iowa is relatively smaller, but strong in research. The Mayo is huge, but the standalone hospital based. It's not like university based. So there's no like other department like literature or philosophy or music. So like diversity is much less. And it's more like clinically focused, especially in psychiatry, is clinically focused. The research was probably Iowa has more diversity and strong research exists in Iowa. And the Stanford, I thought it's like much smaller, but actually, I mean, compared to like a big university, but actually it's big. So I'm still learning. So it's a lot of diversity. Yeah, for sure. So if you look for a job or training in US, I would recommend you to look around carefully to see what will be the best fit for you.
It's wonderful. And considering the collaborators, I was surprised because the first one, John Cromwell, he was one of my mentors too in Iowa, because we had a common project about machine learning.
Oh, okay, sure.
Yes.
So he's a fantastic guy. And so, I mean, he was the one who worked with me for the EEG device. Now we decide not to go together. So he's doing his own thing and I'm doing my own thing. But yeah, he's a smart guy.
Yeah, no, he's wonderful. And that's my other question for you for sharing with the Brazilian students. Considering like these, because you showed us a lot of different skills, no, like animal, human models, new device, epigenetic, and for the epigenetic studies, you need a very strong algorithm like machine learning. You need to learn about computational language as R. I don't know if you prefer R or Python, but you know, so many skills, different skills that these students they need to learn, you know, and yes, you know Pedro, but Pedro has a brother, Vinicius. Vinicius is studying in the high school, and in high school they have a program like a semester that he can make choices for studying a new language like Java, R, or Python. And for the students, what's your recommendation? Because sometimes the students they are struggling, you know, what it's important, it's not important. Or I need to focus more in this, and no, I don't need that, or let the statistician take care of this. What your message for the Brazilian students?
Yeah, so for the full disclosure, I'm not a programmer. And I did a master's degree in bioinformatics when I was at the University of Iowa. So I did learn Python and R and SAS. And I thought I did pretty well when I was doing it. But with older brain, once you stop doing it, you forget everything. But instead I asked my student to do it. So my favorite is Python or R. But my recommendation for the young student would be focusing on one language and be good at it very well. Then application for next language should be much easier. That's what I heard. I only do Python and R only. So.
No, sounds great. Thank you. Appreciate your thoughts. And I'm doing a lot. I have a lot of questions to do, but I know that we will finish this in 5 minutes. But if someone from the audience wants to ask, please unmute yourself or type for me that I can reply the question to Professor Shinozaki. One of the questions that is really important for our post-graduation program is to start a collaboration. And my question is, are you available to accept Brazilian fellows in your department? And if yes, how is the process to do this? To be more practical.
Yeah, so I do not have a funding to support the salary, but if you can bring your own salary, I'm happy to discuss. And in fact, I have been accepting many students, many scientists from Japan. Like most of them have been psychiatrists. So far, I have accepted like 2 psychiatrists from Japan who already finished their times and then back. Then I have 3 postdoc in my lab, and all of them brought their own funding. And another one coming in September, and the 3 more coming in next spring. So we are doing it. So if you're interested to work with us, let me know.
Great. And considering the undergraduate fellows, like because Albert Einstein now has a medical college, a new medical college, do you think that it's possible to do some collaboration considering research? Because now we have this new environment, the Zoom, for example. We can start like working with these guys, and maybe they can go to Stanford to visit you. What do you think?
Yeah, so I mean, the international collaboration is possible, certainly. And actually, I'm doing some with the Japanese institution hospital. So no, why not do is a hospital in Brazil? And if we establish more collaborations, then accepting a trainee would be a possibility.
Wonderful. Wow, I know that Pedro is in great hands.
I have been happy that he is with me. He's a great hand for us. Yeah.
Thank you very much. And you know, I'm learning a lot with Pedro and you, because you are an inspiration for us. And I started to work with undergraduate guys in São Paulo, Brazil, because of you, because of your expertise, your successful stories. Because many times we focus more, you know, in people that are graduated, you know, like medical residents, for example, pharmacists, and you deny these, you know, our attention for the undergraduate people. And I started last year with 3 medical students from Albert Einstein, and for me was amazing. I learned a lot of these guys. We published a couple of papers together because they are really motivated. And I want to tell you that because of you, yes, I didn't know you personally. That's our first time that you are talking together. But I thank you very much. I appreciate you.
Thank you
Being here with us virtually in São Paulo, Brazil. I hope that in the future we can invite you in person in São Paulo for visit Albert Einstein, the medical college, the post-graduation. Albert Einstein post-graduation is doing some movements, and I hope to invite you to give us some talks to participate in our program, because you have a lot of knowledge, like all of your skills in research. I think that it's very helpful for us to keep the journey together in Albert Einstein. Thank you very much. I really appreciate it.
Thank you very much.
Thank you. And I will finish the Professor Shinozaki presentation if there is no more questions. And if there is questions, please send us that I can send to Dr. Shinozaki. We have during the presentation an issue in recording Dr. Shinozaki talk, but we will try to solve this doing some edits, and in a couple of hours we will share for everybody YouTube link. Thank you very much. Thank you, Dr. Shinozaki.
Thank you.
My pleasure.
Bye-bye.
Bye-bye. Thanks, everybody.